And good afternoon, molecular genetics friends. So, I've made a mess of this semester. Cheers. But, controlled transcription, that's my jam. And I thought we would actually just continue on discussing some of the fundamental mechanisms within the control of transcription. And there's some very cool stories here. That is, how genes can be regulated in terms of being turned off and on. This is in one of the books. This is chapter 26. I'm not sure which version. We skipped a bit. Some of the things we skipped was translation, but we've talked about translation in molecular cell. We've talked about genetics, and other things we've skipped were mechanisms behind RNA stability. RNA can be localized. RNA can actually be very long-lived. It can be protected. So gene regulation then can be quite complicated, at least in eukaryotes. In prokaryotes, RNAs are very short-lived. They have half-lives on the order of minutes. So the production of the RNA really is a critical step in the production of a protein. And one of the things that we want to think about is metabolically, the expense of producing protein is quite high. You have to have the amino acid biosynthesis. You're using ATP and GTP every time you're making a peptide bond and moving the RNA, the ribosomes down 3 nucleotides at a time. So the production of protein is something that we shouldn't take lightly. Consequently, it would be better to not have proteins that you don't need right away floating around in your cytoplasm. It's better to actually have some kind of mechanism that says, hey, we need this enzyme right now, start producing it. And we'll see that in the eukaryotes, we kind of play at both sides. And sometimes we'll have proteins that you don't really need right away present. Sometimes there'll be RNAs, sometimes there'll be proteins. Other times we have RNAs that are synthesized just in time. But with bacteria, it's pretty much all just-in-time delivery. So we'll think about this in terms of some of the basics and mechanisms. And as we said, RNA polymerase in the prokaryote has binding affinity to the promoter. Consequently, if we block the promoter, or if we kind of put a roadblock up between the promoter and +1, we can inhibit the progress of RNA polymerase or the binding of RNA polymerase just that easy. Now, in eukaryotes, in some ways, it's actually a little bit simpler because if you don't have the transcription factor binding to the enhancer, the RNA polymerase is not going to bind, and the basal apparatus isn't going to assemble very efficiently, and so you just never get transcription at all. But with the prokaryotic system, if there's nothing in the way, that gene's on. So, that's a way to think about some of the regulatory models. So, a general kind of concept is you have a gene, or a portion of the chromosome. In bacteria, it's a circular chromosome, but that's irrelevant, producing the messenger RNA that produces a regulator protein. That regulator protein is going to have a second site that it can bind to. We use this term of this being a transacting factor in that it is able to diffuse around and bind the target site, this DNA sequence. We would refer to this as a cis element. So that's an important thing that we'll be using in terms a bit, and that is cis elements. These are DNA elements associated with the gene we're regulating, and transacting factors. All right. So a cis-acting system in which we've got the operator and promoter. The term operator is just an old-fashioned term that was used to recognize this is a DNA sequence that is involved in regulation. And when the gene is on, the RNA polymerase can initiate and then generate an RNA. We make a protein. Everybody's happy. When the gene is turned off, a repressor is bound to some kind of sequence. We can call it the operator, whatever. And so this is what is referred to as negative control. We have a transacting factor binding to the cis element, and that turns off transcription. So that's negative control. Now, in positive control, the transacting factor has to bind in order for the RNA polymerase to initiate transcription. Now, in this case, in a positive control, typically what we've got is a gene that doesn't have a great or perfect promoter. It requires a secondary kind of helper in order for the sigma factor to recognize this as a good promoter and to bind to it. And so that's how we can think about a positive control transacting factor. This is some kind of activator system. And there's other ways we can think of that as well. But still, this is positive control. That is, the gene is turned on by activators, not turned off by repressors. All right. Regulatory circuits, these off and on swing things, man, they can be using all kinds of different systems, and we can come up with examples for this. We've got repression, and then the binding of an inducer that causes the repressor to fall off, and we now have gene activation. We also have an inactive repressor binding a co-repressor. It now becomes active, and it represses. Then we've got positive control, in which we have an activator that requires some kind of inducer, and then it becomes a functional activator. Or we have an active activator that can become repressed. So we have a co-repressor. Logic games, there's just a million ways you can build these little Lego sets of gene regulation. So the lac operon is a standard classic model. It was one of the very first that was really carefully studied. Jacob Monod, I think, won the Nobel Prize for this, and that they dissected out the function of the different sequences, developing mutations along the way in a variety of different things. We have mutations that are cis-acting, mutations in the promoter operator regulatory region, and then we have mutations in some of the different players like the lac repressor. So the lac operon itself, as it turns out, the repressor is right next to the enzyme system that we're turning on. It didn't have to be, it just is. So this is the lac operon, and when it's turned on, it produces a long RNA that has the ability, is polycistronic. It codes and allows for the translation of a range of proteins. There's the LAC-Z gene, which produces beta-galactosidase. There's LAC-Y, which produces the permease, and LAC-A, which is the transacetylase. These are two auxiliary enzymes that are going to allow the lactose to get into the cell, and once it gets into the cell, it is acetylated so it can't diffuse back out, and then the beta-galactosidase is able to chew it up, break it apart. All right, so the repressor is produced by the LAC-I gene, and then we have the promoter and operator region. All right, so the promoter is working like a good promoter, and it is a pretty good promoter. There's a few issues with it, but we'll see in a moment. And then there's the operator, which is the binding site for the repressor. So the operator is binding right around here. The promoter is binding around here. And this is going to, when the operator's bound, it blocks the expression of the gene. So the inducer, which can be lactose or essentially an artificial sweetener that we use, which is a non-hydrolyzable version of it, which causes the repressor to fall off, and it allows for the production of the protein. Now, if we look at the messenger RNA, when you add the inducer, it comes right on up, and then it begins to go down fairly quickly. The protein itself, though, is a good bit longer lived. It takes a few moments for the protein to start really coming up, but then we have protein present for a while. So the RNA is short-lived, but the protein product actually hangs around for a while. And this is how the lac repressor operates. It actually binds in a cooperative manner, and it binds primarily as a tetramer. So a lot of mutations have been carried out identifying different parts of this protein that are involved in forming the tetramer. Let's see. So if you add the inducer, the sugar, it causes the repressor to fall off. Now, there are a couple of different mutations that are interesting. There are operator mutations that you change the sequence such that the repressor can't bind, and consequently, beta-galactosidase is now on constitutively. And so this is one of the ways that the mutation system and the whole repressor system was worked out by developing these kinds of mutations and things. Now, mutations that inactivate the LAC-I gene cause the operon to be constitutively expressed as well, because the repressor can't bind to the normal operator. Now, just as a little hint, experimentally what you would do to decide whether you have a mutant repressor or a mutant operator is you would introduce a plasmid that has a functional repressor on it. You express that. If the system is repressed, that means you had a mutant repressor in the genome. If the system isn't repressed, even though you've got guaranteed a known functioning repressor, that's probably an operator mutation. All right, so we had these different domains and crystal structure. All right, but here's what it is. We have the DNA binding domain, and then these different kinds of isomerization or tetramerization domains and things. Mutations have been identified in this, in that you chop this part out and it becomes a recessive in which it can't repress. Now, if you chop that part out, it becomes what's called a dominant negative, and that it can't bind to the DNA, and so it is never going to be able to repress at all. And you can chop out this part or make mutations there. It's dominant. It either can't bind or it can't respond to the inducer. So the LAC operon the sequence is known, and this is one of the very first DNA sequences that was actually generated. And what we have then is the interaction of the repressor with the DNA, creating this sort of kink structure here, and then that kink is relaxed when the inducer is present. Okay. Is there anything really all that useful here? Not really. So the repressor binds at a pretty good affinity, specifically compared to other DNAs, and its specificity is quite high. When the operon is induced, the operator goes from being 96% bound to 3% bound. It falls off. So at any one point, pretty much all of the repressor is bound to the DNA. Now, a small molecular inducer, that is cyclic AMP, is able to interact with a protein known as catabolite repressor protein, or CRP. And this is what's going to bind to the promoter and assist the RNA polymerase in initiating transcription. So this is an example of where the promoter is not very good, and it requires, in order to fire really at high level, this auxiliary thing. And what this does, here's a picture of cyclic AMP, nothing we care. What this does is allows for inhibition of this system if both lactose and glucose are present. So if lactose is present, then the system turns on. If lactose is absent, the system is off. If lactose is present, the system and glucose is present, the system would rather eat glucose than lactose. So in the presence of both glucose and lactose, the LAC operon is not repressed, but it's not firing very well. Now, if glucose levels begin to drop and lactose stays high, there's going to come a point where glucose levels are too low, and that induces a stress response that results in the production of cyclic AMP. That's going to in turn bind to this catabolite repressor protein and now allow for effective transcription of this RNA. And this RNA is actually, or this binding of the CRP is found in a variety of different kinds of places. All right, we're going to stop here for just a moment. All right, picking up where we were. a momentary break. Another really interesting gene regulation system involves tryptophan biosynthesis. And there is something known as the trip repressor. And it recognizes genes, or a couple of different genes that are involved in the tryptophan biosynthetic pathway. Now, the trip biosynthesis system is controlled in part by termination. You can have early termination or you can have read-through. And the termination is controlled through changes in the RNA secondary structure, and they are in part determined by the ribosome movement. So here's how it works. We have what's known as the leader sequence, which is the initial open reading frame. And the leader sequence actually has a series of tryptophans that are codons that code for tryptophan. Now, one of the things that's kind of interesting to think about is, let's imagine we're a bacterium and we get low on tryptophan. We need to induce the tryptophan biosynthetic pathway. Now, on one hand, you might say, wow, if you're low on tryptophan, the last thing you want is to have a protein that requires tryptophan or some sort of regulation that requires tryptophan. But this is kind of a thermostat in that you don't want to let it get too low. You have to have it at a moderate spot where, okay, we're getting low, we're not at dead zero. And this is a clever way of doing it. So if tryptophan is at low levels, there'll be a momentary sort of stutter pause when the tryptophan codon is called for. So the amino acids that are on the tRNAs are going to be more scarce. So what happens is the polymerase initiates and it produces this RNA. Now, if tryptophan is absent, if we have low levels of tryptophan, then the polymerase continues on and we have translation initiating and we've got the biosynthetic machinery. But if tryptophan is present, here's what happens. The system forms a hairpin here, and that causes the RNA polymerase to actually fall off. So if tryptophan is present, the ribosome can start moving up this thing fairly fast. And so here's an example of the leader peptide. You've got a methionine, initiating methionine, these two trips here. Now, this is involved with the formation of a hairpin as well. And we can have a couple of different structures for this termination hairpin. And this is looking at it for just a second. So here is the trip leader region in a couple of different formations. It can form a termination hairpin, but then we have these alternative structures in which we don't generate a terminator sequence or a terminator structure, and it allows for read-through. Here's a better picture of it here. All right, so if tryptophan is absent, the ribosome is going to stop at these trip codons. It's going to hang there. That causes this structure to form. If tryptophan is present, the ribosome pushing forward disrupts this pairing, and now you have this pairing, and that forms a really potent terminator hairpin. So the presence of tryptophan and the absence of stalling changes the structure of this thing. Pretty cool, yes? It is. So in the presence of trip tRNAs, the ribosome translates the leader peptide and it's released, and they are able to form this terminator. In the absence, though, the ribosome continues up and you begin biosynthesizing it. So now, this same kind of system is actually seen with a bunch of other amino acid biosynthetic pathways in which you have a leader sequence, and the peptide in that leader sequence is going to call for the amino acid that it's trying to biosynthesize. And so this stalling effect can change the structure of termination. Now, this is a really interesting kind of model, let's just think for a second why this is not present or possible in a eukaryote. And the answer is, of course, there is no co-translation with transcription. So in the eukaryotes, translation is completely separate. Transcription occurs in the nucleus, the RNA is processed, then moved out into the cytoplasm where it's translated. So this sort of system, as clever as it is a complete non-starter for a eukaryotic system. All right, guys, we're going to knock off for now. We'll pick back up after next week and cover a few more bits and see where we go. Thanks for your attention. Hope you're watching. Rock on.